Distinguished guests, ladies and gentlemen, good morning, everyone. First of all, thank you, the organizing committee, for having me. It's my pleasure to share with you for platinum-based drugs. What about the latest progress and the prospect going forward for the future as well? So I got entrusted by General Manager Wang to talk about my presentation in a dozen minutes. So in my time session, I'm going to walk you through about platinum anti-tumor drugs. What about the process, status quo, and development history? So please feel free to correct me if you think I'm not very comprehensive in presenting. So I prepared some slides, and the concluding few slides is going to look forward into the future as to the precious metal. So Commission Zhang and also the Zhu both mentioned that PGM has unique attributes in the biomedicine. What about the much anticipated and much look forward to research results that we can embrace as well? Think about in the area of a drug. Metal is not the most important one. As is known to all that, small molecular is mostly based on the organic chemistries. And some newly added ones like, for example, think about a protein-based antibody, large molecular drugs. These are the new forefront of our technology development. They don't want to use or involve any metallic elements. But let me assure you that, as a matter of fact, in some aspect of drug and treatment, so there are some important ones, actually, which will allow for a pivotal role by the metals. So they are irreplaceable. So we have a diagnosis of drugs. So we have a radioactive, like the epitope. So metallic elements. For the early discovery of the diseases, they played a very important role. As a result of that, they can provide a lot of unique properties not owned by others. At the same time, we can use precious metal for treatment as well. So you'll find that genetic and also the gold-based, platinum-based are being used one for development of the anti-tumor drugs, and there are a lot of drugs being worked on as well. So the most leading case in point is a platinum-based compound used for treatment of cancer. Cisplatin, as known to all that, it's a serendipity. It happens by coincidence because the researchers are trying to apply the platinum-based electrode, and they are trying to apply it in the cellular organizational environment. They are thinking about studying the E. coli under the magnetic field and electronic field. What about the variation of the E. coli? Turns out that it's nothing related to the magnetic and electric field, and then the platinum-based electrode in the solution. As a result of that, so there is a highly active chemical, cisplatin, is being generated in the process. So accidentally, we discovered the cisplatin. So think about it was discovered first in 18-something. So think about in the process of heterogenation, and so cisplatin is one of the most prominent ideas being discovered. So what about the cisplatin is very effective at a clinical level, but not true for transplanting. And after more than a decade of efforts, very happy to see that. So traditionally, something which is turned away by the scientists, like heavy metal, are being applied in a clinical trial. So think about the cancer patients when they go through chemotherapy. So especially cisplatin, a platinum-based compound, has played wonderful, miraculous roles in helping to alleviate the symptoms of the patients. Think about the famous athletes in the world after being diagnosed with the cancer, and they have gone through the surgical operation, and then they use platinum-based chemotherapy. So they recover their health, but also they can go back to their career life to become excellent athletes, win a lot of awards as well. Well, from that perspective, so think about the cisplatin. In terms of saving a lot of patients, they used to play a very important contribution. So let's try to look at the history of cisplatin ever since its discovery all the way to the year 2007. Throughout the history of the platinum-based drug discovery and research. So these are the milestones. I'm aware of the time we have, so I'm not going to go through things one by one, but I'm going to share with you that ever since year 2007, why is it the case that we didn't see any new platinum-based drug as being approved by FDA? So actually, whereas no more newly approved platinum-based treatment regime anymore. So I think we have a lot of highly active ones, more active than cisplatin. We have a carboplatin and oxaliplatin, etc. But they didn't make any inroads or breakthroughs at the clinical level, but we never stopped our efforts to do the R&D for them. So now, this is about the milestone and roadmap for the chemotherapy. In the areas of clinical treatment, these are the compounds being used as well. So you can clearly see that. So you find that actually carboplatin, cisplatin, and oxaliplatin, these are the three approved platinum-based treatment regime approved by the FDA. So for China, Korea, and Japan, there are a few platinum-based ones. Together with the three approved platinum-based ones approved by the FDA, when they are being used in combination, they can play wonders, work wonders. Now, ladies and gentlemen, so let's try to look at the AAA highest rated hospitals. What about the clinical cancer patients they're treating, and how are they being treated? So as a matter of fact, we understand that chemotherapy is being widely used a lot, especially in the context of where people have a higher awareness to accept the chemotherapy. Chemotherapy is something we must embrace for the serious patients, especially if the patients are in the process of the development and growth of the tumor. So chemotherapy can play a very good inhibitive role. Just because of that, happy to see that now in the market, roughly 0.5% of the chemotherapy regime will have to be based on platinum, all different types of platinum things. So the platins. So as I mentioned, so the ovarian and also, for example, the bladder cancer and also the head neck cancer and triple negative breast cancer, and those ones actually are going to use a cisplatin as a first-line treatment. Something worthy of sharing with everyone is that over the past 10 years, we understand the chemo and the radiotherapy over the past decade has already become the forefront of the first-line treatment therapy at the clinical level. And immunotherapy used in combination with platinum-based chemotherapy has worked wonders in terms of treating intractable diseases for a lot of difficult patients. So that is to say that platinum-based drugs under the current context of a combination between the platinum-based chemotherapy and immunotherapy. So we can use those two in combination. As a result of that, a combined therapy is going to play a bigger role. And also they can address the issues of side effects like toxicity and enhance the efficiency, efficacy. This is very helpful. Cisplatin is problematic in the sense that its toxicity and the drug resistance actually, for example, is a key issue. So people are thinking that maybe this is not an effective cancer treatment therapy. And also from the molecular design perspective, a chemist is going to think about, is it possible that we find something compared with the cisplatin, carboplatin, and also oxaliplatin, we can find some higher active ones than them with lower toxicity profile. This is something we are always constantly pursuing. So that's why over the past 4 decades, you find this area has been hotly pursued by international world-leading chemists as well. And even since the lifting of the COVID restriction measures, the US and the UK, you find Medical Research Council together with FDA have jointly published the 9 challenges for the treatment of cancer. So they are trying to propose the idea of how we can think about trying to get rid of the issues caused by toxicity by using the carboplatin, especially the neurotoxicity is a huge challenge. They're trying to propose that one of the 9 challenges that they are trying to solicit a worldwide scientific contribution as to how can we make breakthroughs for that toxicity for carboplatin. So maybe the answer is within the hands of a chemist. So maybe new molecular structure powered by the precious metal PGM is going to play wonders. And the most clinically viable ones are the optimization of the platinum-based compound. So Commission Zhang and also Li Jun also is asking the partner of Commission Zhang, given so many compounds, why is it that only platinum-based ones or the platinum-based ones can be used? So there's no single answer to that because we all understand that for example, cisplatin, it works its wonders and the working mechanism is understood, and so that it will induce the apoptosis of the cancer cells. But in the periodicals of precious metals, so think about actually given the similar spatial electrical structure, there are so many different elements in the periodicals similar to the platinum. Why only platinum can have the chance to be used at a clinical level? So we should use a deep dive dive into theoretical understanding in order to understand what we can do. So the answer is within the hands of the chemists. We have to make some further efforts to do the research. So from the platinum, can we make sure that from the nucleotide to the granulocyte, we understand without the protection of the proteomics, and it will be easily be susceptible to damages. So if that is the case, so maybe we are thinking about in addressing the drug like resistance or toxicity. Maybe this is going to be helpful or not or something. So you will find that actually. So in terms of regulating the like energy metabolism and regulating the glycolysis is going to be helpful. And also there is a problem of a mitochondria regulation mechanism. Maybe this can be helpful as well. And also nanoparticle can help the reparation and also repairing in an intelligent way, and also to try to have a visualizable and a visible approach to release only the effective part of the platinum without releasing the other part, which will cause the problems. So let me give you the example of one type of molecular structure which I think stand a chance to make sure that platinum-based solutions can be designed in new ways to find its new root picture in its clinical trial applications and used in combination, and how are we going to let the platinum-based ones work its wonders. So these are some examples that I really want to give you. So from the like double valent to the tetravalent platinum, by changing the valency, I believe that. So think about actually. So the PD, PK is going to change a lot. So think about, let's try to look at the cisplatin. So think about the double valent. So think about within 2 hours, a lot of hydrolysis will happen. And also for the patients suffering from pathogens, maybe like the hydrolysis compound from the cisplatin is going to be effective. If you turn from the double valent to the quadruple, which is a tetra, like valent, and then so it could be there existent for several weeks without having to change its structure. So this is a very important dynamics, for example, science for you. Within the living organism, so there are a lot of different like reductant like glutathione. So if we are trying to work back from the tetravalent to the double valent cisplatin, so maybe we can let it combine with the big molecular science to make sure we can regulate the change of the down-regulating signal and trying to induce the apoptosis of the cancer cells. So we call it a pre-drug or prodrug. Prodrug or pre-drug molecular will work wonders. It's very helpful for axial, like horizontal lines. We're going to use like the platinum-based ADC to anti-drug conjugate, and then from the axial line. So maybe how can we try to integrate it with some exogenous functional groups, and then we can integrate a lot of different functions on the same set of molecular structure. So on this trajectory, people are making a lot of attempts. I'm aware of the time I have. I'm now going to elaborate ideas. So from the molecular design perspective, this is about what's going on and how can we do the optimization. And also from the clinical doctor perspective, and how are we going to address the above-mentioned issues. So think about the issues and the side events, and also the adverse events for the like platinum. So maybe we can use the chemotherapy plus the RT, CT and RT radiotherapy. So the non-small cell lung cancer and gastric cancer, so they are standardized first-line treatment, but you have to include carboplatin or cisplatin. So platinum has to be there. And going forward, I'm going to share with you more additional pictures. So trying to look at the like immunotherapy right now. In the current context, it's being used a lot. It works effectively, but the key issue is about the response rate is not high enough. So response rate is only 20%, which means that only 20% of the patients after subjecting to the immunotherapy can benefit from it in any ways. So the cisplatin and also the immuno checkpoint inhibitor, if it is used in combination, we can make sure that it will work in synergy together. So actually, instead of letting them being combined together seemingly, but they still work separately. So how do they work in synergistic ways more effectively by putting them together? These are the new research directions. In the afternoon, professor from Beijing University, Liu Zhibo, is going to focus on how can we redesign the molecular structure to make sure that RT and CT can be combined together. So This professor mentioned that for a series of tetravalent compound based on the platinum, based on iteration, so they can become actually the hydrolysis and also free radical electrons. This in a very efficient way to make sure that a pre-drug or pro-drug for tetravalent platinum-based solution, based on some conditions, they can effectively release the platinum content. So think about this is about oxaliplatin. Happy to see that so in such a context, so this effectiveness and efficacy in vivo and in vitro, it work wonders. It's very effective. And going forward, attempts like that is going to be very viable at a clinical level. It's because our radiotherapy and RT actually is going to be applied a lot by doctors as well. So maybe this is like a very generic treatment regime going forward for X-ray. So it's not being affected by the penetration depth. X-ray is different from other different rays. Say it's very miraculous. So think about like the photosensitive drugs, when it's being ending up in the bodies of the patients, it comes with a lot of constraints. But RT combined with a CT like this way doesn't have that problem. CT is a chemotherapy and RT is a radiotherapy. And so we are going to make continuous attempts on that front. Hopefully we can make some inroads at a clinical level because both treatment methods are being approved by the hospitals as well. So the next issue is that, given the currently effective immuno checkpoint inhibitor drug and compared with a traditional viable platinum-based chemotherapy, how can we let them work together? So what is that actually a platinum-based ADC doesn't make any breakthroughs? I think one of the roadblock is something people are keeping an eye on. So there needs to be enough payload. So think about actually it's like the probe. Probe needs to be toxic enough and the payload should be toxic enough so that based on the high selectivity of the antibody, if those two are being ready, we can effectively combine the two drugs. But the problem has already come. Is it possible that maybe we can use the high selectivity of targeted points for the antibody for certain targeted points and trying to let platinum play its role beside and beyond the chemotherapy? How can we let platinum work its wonders? Put it in simple terms, is it possible that for the chemotherapy, so the platinum payload can be reduced significantly? Is it okay? So can we both combine the beauty of the two? Do you think it's possible if we can do both? Can we address the issues, the toxicity issues of the platinum-based drugs or drug resistance issues? Can it be addressed as well? So drug resistance and toxicity issues there is because we use a lot of unnecessary, ineffective, heavy overdose of platinum there in places where it's not needed. So it's not highly selective for its absorption and it's for intake. So think about if we can combine the platinum-centric solutions with antibody, maybe there will be some new working mechanism like ADC that will be worked out as well. And also a second note is that for the specific metallic component and compound based on the platinum with special features, so we have to improve our clinical screening method as well, because think about a lot of ineffective cellular killers are being there in the mixture, but effective ones are being screened out. So this is not going to be okay. We work with the Global Hospital in a sense that we are using the tetravalent platinum and also trying to combine that with a PD-1 immuno checkpoint inhibitor and using very minimal low dosage of platinum. Turns out that a very interesting will happen. The results is very interesting. So think about the T cell function has been enhanced. Platinum drug, as you know, that is going to minimize or compromise the immune capabilities of the patients. But quite coincidentally, we find out because of the use of the platinum-based solutions, the immune system has been beefed up for some patients because of the using of the platinum. So going forward, if you screen for viable versus non-viable compound, maybe there needs to be some new screening method as well. Last but not least, before I conclude, some personal contributions, if I may. I think going forward, so under such a context of the new strategies, I believe that the platinum is going to have a new vitality as well. So let's give you the example of platinum-based ADC drug. So it's not just 1 + 1 = 2. Maybe we hope that a synergistic effect by working together is stronger can be made available. And secondly, for the platinum-based drugs. So actually, from the perspective of chemistry, this is not about a compound polymerization of organic molecules. So there are a lot of the thermodynamics and also the electrochemistry at play. So we need to embrace new screening method for the validity of the drugs going forward. And secondly, we have to think about platinum-based drugs. Toxicity issue needs to be addressed as well, especially the one confusing a lot of patients, like neuron toxicity. So think about, I believe that this will build up. It will have cumulative effects. If you use it for a certain while, there will be the side effects and adverse events for the patients. At a very basic layer, so think about NIH. And also the UK's Medical Research Council, they're trying to issue the call for addressing the 9 pain points and challenges for toxicity and the resistance of the drugs. Also think about actually for different cells, so T cells and tumor cells for the platinum for its distribution. In the process, what about the similarities? What about dissimilarities? I think I think these are the worthy areas of our further research going forward. Last but not least, do you think that is a viable strategy for us to make sure that, given the patient, so let me give you one example. If you use a radioactive to do the diagnosis, is it possible that we can follow from the intake of the drug all the point throughout its cycle for the drug to finish its mission? Is it a way for track the drugs throughout the cycle for the platinum-based solution? Now, it's still in a black box. We have no idea as to how it works and when exactly it works. So, like cisplatin, when it goes into the nuclei of the cells, so actinopoietin by Madame Zhu is going to induce apoptosis or something prior to arriving at the core of the cells. What else? Where else could it be bees to work wonders as well? And also to generate the beefing up the effect of the immune system before the reaching of this new cell nucleus. Where else could it work effectively as well? This is not a summary because this is just food for thought for the future. So we very much look forward for some new mechanism for the platinum-based drugs. There may be some new mechanisms as well. So I think aside from this one's MHC, so for example, very important immune protein in our system. Aside from that, so when platinum ends up in the nucleus of the cells. So after doing the lysis, so what about the possibility of generating new antigens based on the platinum? So the new antigen, what kind of role will that play for treatment of cancer? Nobody tried to understand that yet. But I think it's a worthy direction for our further research efforts as well. Okay, ladies and gentlemen, before I conclude, I would like to say some acknowledgement. Thank you, my team, working together with me over the past few decades. Thank you very much for the great pivotal role you have played in our industry, especially collaborators, so coming from the Bell Research, Medical Research, and Animal Research Lab, etc., and also radioactive therapy. And we work with Dr. Liu Zhibo from the Beijing University for the Life Science dedicated session. Liu Zhibo is going to make some keynote presentation. Let me conclude, if I may. Ever since about 3 decades ago, ever since we tried to work on using platinum-based one to do the tumor treatment, I have heard a lot of doubts being raised about this area. People take it for granted that for the platinum-based drug, people think there's no future for that. And generally speaking, so people are saying that chemotherapy-free or chemotherapy free kind of cancer treatment. But as a chemist, since the beginning, from the molecular design perspective, and I think it's my liability and responsibility to find out a better mechanism of action from the molecular design perspective to try to find a better option for platinum-based solution to address the baffling issues right now, to find more progress for the clinical application of platinum. Okay, thank you.